Good morning. We are having people logging in, so we are going to wait a few more minutes.
Good morning.
Good morning, everyone. We are maybe 1 or 2 more minutes to see if we have more people logging in. We have 25. Okay, I think I'm going to start welcoming you, all the attendees. We are still having people logging in, but let's just start with a few words. This is the 6th year in a row that we have CABSCon. CABSCon is organized by the CABS, the Center for Applied Biotechnology Studies in Cal State Fullerton. A few years ago, we decided that we were going to privilege the organization of a conference on biotechnology as one of the activities of the center. And well, it has been successful. 6 years that we are having it. Unfortunately, this is the 2nd year that we go online. Hopefully next year is going to be back to presential. So We really miss mingling with people, miss having breakfast together and then stop and having lunch together in between talks. We hope that this is the last time that it happens like this, and next time we are going back to that. Dr. Andrew Tomaras was going to be the first speaker, and he just told us that he has an emergency and he cannot speak. So the organizing committee decided that we are going to replace that talk with another one, and that is me. So you are going to hear a talk by me as first conference. A few housekeeping things. We are going to have the conference, and then we are going to have a period for the poster session. I think everybody is aware. You can go into Slack to read the poster. That would be the equivalent when you are in a conference and you walk around and the poster is placed, but there is no people manning them. But you are free to go and look, and that was actually open since yesterday. If you want to ask a question to the author in Slack, you can do it by just sending a message through Slack. You place the question in there and you'll be answered. But also you have a Zoom link for each of the poster presenters. And if you go into that Zoom, between 10 and 10:30 and 11:30 and noon, they are going to be there, and then you can hear the presentation of the poster, or you can just simply discuss with the speakers. Then at the indicated time, you come back to the main Zoom, and we continue with the talks. So let's hope for a productive and good meeting. And I'm going to just directly go, since I'm going to be the first speaker replacing Dr. Tomaras. I'm going to start talking, and then when I'm done, we will have the first poster session. So I'm going to share the screen. Let me see, it's 9:06, and we are still having people logging in. So maybe it's a good idea to wait 2 minutes, but I'm going to start with the introduction, and then if somebody logs in, they can just follow the talk. So I'm going to share the screen. So we are going to talk about not the pandemic that is making us having this meeting online, but we are going to talk about another pandemic, the silent pandemic. Something, as you are going to see, I have a slide where people consider that the next pandemic is what is coming, that is antibiotic resistance. So I'm going to start by just giving you a quick description of the impact of antibiotic resistance and the way that we approach that in our laboratory. So the first thing that we can talk on antibiotic resistance is simply the infection. You are in the community, you catch an infection and you get sick, or you go to the hospital to be treated and you get an infection in the hospital. And if the infection is resistant to antibiotic, it can be problematic. However, there is another aspect that is very important. That is not just the primary infection, is the importance of the antibiotics for medical procedures. So if we start having resistant strains, we are going to lose the ability to just do regular surgery, prosthetic replacement. And I already could talk myself for 2 or 3 cases close to me that they got hip replacements, and then they get an infection that is not responding to antibiotics, even dental work. So there is a lot of medical procedures that are dependent on keeping infection out. So that is a tremendous problem. When we consider the resistance to antibiotics in regards to all of these issues, in our laboratory, we just attack the problem from many aspects. Almost every aspect you can imagine. We are interested in the genetic organization of the resistant genes, in how the genes disseminate, how they resist, the actual mechanisms, and then the molecules in charge of producing the resistance, like the proteins and so forth. And this is a conference on biotech, and biotech also, you want to find solutions. We also try to find a solution to the problem of resistance to antibiotic, which means if we are losing antibiotics, how we recover them, with new antibiotics or with some tricks that we can do to be able to use existing antibiotics, and that is inhibition of the resistance. And there is another aspect that is not something that happens in the laboratory, but is very important, that is the issue of the increase of resistance in the environment due to behaviors, and sometimes are behaviors that are just simply because we don't know that we could do better, and sometimes because we don't know, we do things that just increase the amount of resistance, or sometimes you know that it's happening and it's inevitable and you have to do it. So a few of these things that really we can do, and we are doing in some cases a lot to avoid, is to educate people not to self-prescribe, to comply with what the doctor says, not to use it as additives, not to feed animals for human consumption. That is one aspect that we are doing good, because we are not having so many animals for human consumption being fed antibiotics. And also very important, when the industry sometimes is they are producing antibiotics and they dump the waste, and sometimes the waste is loaded with antibiotics, and then suddenly you are filling the environment with that. That is within the realm of the public health. And that is something that we are also interesting. So we call the left of the slide conventional, what we do in the laboratory, or at least for us, what I've been doing all my life in the laboratory. To the right, public health is something relatively new for us, and we do it through a program that is a mix of teaching and research. We train students through the Minority Health and Health Disparities Research Training Program. It's solely international in the past. Now it's a mix of national and international. I'm not going to talk about that. If somebody is interested, can ask. But all these public health issues are channeled through that in our laboratory. So nowadays, I don't need much to convince people about the importance of antibiotic resistance. It's in the regular press, everybody reads that in the newspapers. So just as a mode of illustration, two important reports that came out recently, one from the United Nations, where they predict this number of death year, and they predict 10 million deaths by the year 2050 if things keep going at the pace they are going. So we better get our hands busy and try to solve the problem. The CDC also produced an important report lately, and it has one important point with respect to our research. That is, they recommend not to rely on new antibiotics. If we are expecting to get new antibiotics to replace those that are lost to resistance, we are not going to do good. We need to find some ways in addition to that. And that is exactly what we do in our laboratory. And this is a very recent slide I put together because in the past 1 or 2 weeks, newspapers all over the world are talking about the next pandemic. So the next pandemic, sorry. The next pandemic is considered, that doesn't mean we will have other things, but this one is coming. That is, we are seeing more and more bacteria that were easily treated in the past that now resist antibiotics, are very complicated, in some cases resist all the antibiotics. So that's a problem that we need to pay attention to. So how is that we got into this subject? This was something that happened long years ago, when in the neonatal wards in children's hospitals, this is particular is a study in the Children's Hospital of Buenos Aires, Argentina. When there is an outbreak of infection in a neonatal ward, you need to treat. You don't have time to do an antibiograms and decide what is the best course of action, although you can do it. but you need to treat immediately because by the time you know what is the best course of action, you have a lot of babies dying. Amikacin is an antibiotic that came into the market in the late 70s and was a great solution for that strategy. Amikacin, when there were outbreaks of infection with Enterobacteria, in particular, you are seeing a case with Klebsiella, it would just always be successful at the beginning. So you knew that you were going to save the lives of the babies treating with amikacin. And you can see that in this year, the level of resistance was 0%. So you knew that it was going to be active. 8 years after, 60% or more of the outbreaks were resistant to amikacin, and that means the babies were dying. And actually, the strains we use are coming from babies that died. So still, there is not a great replacement to amikacin. Some new antibiotics are coming out lately, but still is a big problem. So what is amikacin? I mentioned amikacin is a semi-synthetic antibiotic. That means the natural antibiotic has been changed in the laboratory to produce a new one. And the original is kanamycin, which is an antibiotic of the kind of the aminoglycosides that if you look now at the situation with this antibiotic with respect to resistance, you're going to see that I'm putting different arrows, different colors. Every arrow is a family of enzymes that can inactivate the antibiotic. And you can see that you have many families that do that at different locations by doing some chemical change at different locations of the antibiotic. And the different colors means that the hydrogen is replaced by a group, an acetyl group. Another color means that it's a nucleotidyl group. Another color means it's a phosphate. So we see we can phosphorylate, acetylate, nucleotidylate this antibiotic and inactivate it, and there are almost 100 enzymes that can do it. As a consequence, kanamycin is practically not used anymore in the clinics, but Bristol in the 70s modified kanamycin by adding a chemical group, and amikacin became non-substrate of those enzymes. So it was, at the beginning, it was a big success. It's still a big success because all of these resistance enzymes were unable to work until a 6' acetyl transferase was able to modify it and produce resistance. So this acetyl transferase, what it does is from acetyl-CoA, takes the acetyl group, acetylates, replacing one of the hydrogen. This compound is no longer an antibiotic. We have a lot of related enzymes. This AAC 6' means aminoglycoside acetyl transferase 6', indicating that this is what it does. This 1B is a unique identifier. By today, we have 1A, B, C, D, all the alphabet and double letters. However, in the clinics, 90-something% of the cases that are resistant to amikacin are due to this enzyme, which is within the situation that is not good because amikacin is being lost because of the enzymes. It's good that we have one main enzyme. If we can do something with the enzymes, we would solve a lot of the problems of resistance. So a few characteristics that make this gene interesting and sometimes complicated to work is the more relevant. That is good, I already spoke. But we have variants, and this is something I'm not going to get into now, but if somebody's curious can ask after the seminar, I will discuss it. And the gene can be housed in plasmid, chromosome, gene cassettes, pseudogene cassettes, integral SER modules, the very new SER modules that we know that now that exist, and as a consequence has a very high ability to disseminate among bacteria. So it disseminates among all the gram-negatives. So because today is biotechnology, we are going to concentrate on finding a solution to the problem of resistance. And how can we find the solution? it would be great if we can inhibit the expression of the enzyme. If there is no enzyme, there is no resistance. Or it could be great if we can inhibit the activity of the enzyme. If there is no activity, then there is not acetylation. So here is the translation of mRNA, the protein, the reaction that it catalyzes for resistance. And I'm going to call your attention to these strategies to stop expression of the enzyme. The enzyme doesn't exist if we can stop this process in here. We try to do that with antisense technologies, which are very varied, let's say high variety of technologies. Of all of that, we concentrate on two of them. One is we call steric hindrance, the other EGS, and I'm going to show you a little bit the concepts of this and then a few experimental results. So we have the gene that is transcribed into RNA, translating into the protein that will confer resistance. So we can imagine that we could interfere into the translation. This is the ribosome that has been placed. We can imagine that we can produce an antisense molecule that interacts with the RNA and interferes with the establishment of the ribosome, or if it is down the road, it can interfere with the progress of the translation. And that is what we call steric hindrance. There is another one of the most popular antisense strategies, that is to produce a DNA antisense, or a molecule that looks like DNA. And in this DNA RNA duplex, the endogenous RNase enzyme will find that as a substrate and will produce an endonucleolytic cleavage, and the messenger RNA will be degraded, as you can see. And then another one that is conceptually equivalent, we have the RNA, and we produce an antisense that is an RNA, or a molecule that looks like RNA. And this duplex RNA with the right structure will induce the endogenous ribozyme RNase to produce an endonucleolytic cleavage. And that is what we call EGS technologies. So since EGS technologies is a subject of one of the subjects of this talk, I'm going to talk a little bit more. In here we have a pre-tRNA molecule. This transfer RNA is ready to be mature, and it's going to be matured by the ribozyme RNase. The RNase will recognize this region of the molecule and will produce an endonucleolytic cleavage, and we are going to have the tRNA ready to go and participate in protein synthesis. So this three-dimensional graph, I'm going to project it in 2 dimensions. So now we have the pre-tRNA, the RNase, and the place of cleavage. And what I'm going to do in this slide is I'm going to modify the whole slide, but I'm going to leave this untouched. And the modifications imply that I produce an RNA molecule and I put an AUG to indicate that this is a genetic messenger RNA. And we have another molecule that produces in the interaction a structure similar to the one that had in the pre-tRNA. And I have to tell you that I didn't change the sequences for clarity, but these sequences can be any sequence. The important thing is that the structural aspects are equivalent to the ones that you find in the pre-tRNA. If you achieve that, then you can induce RNase to degrade the messenger. If you can do that, this red molecule acquires a name that is external guide sequence, EGS, and the process of trying to kill a messenger RNA, thereby impeding expression of a gene, is called EGS technology. And that is what we did. And this is the RNA. I introduced you to the messenger RNA of the AACC program 1B, the resistance enzyme. So this mRNA was mapped using biological and bioinformatic techniques in order to determine regions that are amenable to interact with antisense molecules. And there were 5 regions that were clearly identified, and if you put an antisense molecule, they will interact with these sequences, and then you can achieve the duplex that is needed for the RNSP to do the cleavage. So since those regions have a certain number of nucleotides and the EGS has a limited number of nucleotides, for every region you can imagine that you can design a number of EGS molecules. So we did a battery of experiments. All of them follow this strategy. The messenger RNA is labeled, you put the EGS to be tested, RNSP that we produce in the laboratory, and you look and you see if there is a digestion or not. So what you see in this gel is the analysis of the selected EGSs that were successful. So we tried a lot of EGSs. So it's like 10 EGSs per region, which would mean like 50 reactions. This would be a gigantic gel, but the best ones are shown in here. So these 4 lanes are controls. So no RNSP or the random EGS and so forth. And what we see is that the messenger RNA is untouched. But when you put the right EGS that goes to the right place, you can see that each one of these experiments with an EGS targeting a different region produces a fragment after the endonucleolytic cleavage. So these EGSs are the candidates to be able to interfere with expression of the protein. The question is, that is in vitro, how can we do that in vivo? And that is very problematic. So in order to do the proof of concept, we did something that you cannot do in real life, but you can do in the laboratory. That is, produce a special recombinant clone if somebody is interested in how to do this recombinant clone that is very unconventional. The recombinant clone will produce endogenously the EGS. So we don't need to put the EGS from outside into cells, so we can have cells producing EGS. So we created strains that are E. coli that have the resistance gene, and we transform them with plasmid that produces EGS. So every experiment was done with an EGS that is the one that is being analyzed, and 2 EGS that are controls. 2 controls are required in this kind of experiments. One is completely random, an EGS that doesn't interact with anything. One is an EGS that interacts with a gene that has nothing to do with the experiment, and you expect that these EGSs don't do anything. So then we culture these cells in the presence of the antibiotic amikacin. Since they are resistant, you expect them to grow, and this is what happens with the controls. When you put an EGS that is active, they don't grow. Amikacin was able to kill cells that are resistant to amikacin. So this is an experiment that shows that, at least in concept, the EGS technology could be a way to solve this problem. However, when you start thinking, there are lots of challenges, because you cannot generate an EGS. You cannot tell somebody who is sick to get sick with a strain that has a plasmid that generates an EGS. So you need to add the molecules from outside, like with every medicine that you take. So when you start thinking about all of the problems to solve, the list is long, and this is just a partial list. So for now, we are going to concentrate on 2 of these challenges. One is, if we are going to use an RNA, RNA is very unstable. How do you stabilize an RNA to put it in a formulation and people go and buy it and take it? And then when you internalize it, the RNase is in the body, the RNase bacteria will kill it. So we need to solve the problem of stability. And also bacteria normally will not take it up. So you can take a lot of molecules. If bacteria don't take it up, you are not going to cure the person. How do you deal with this first? What you do is you use analogs, and there is a lot of interest in this. A lot of laboratories work only in creating new analogs all the time. So in here I show just sample of the analogs that are available. For example, this was generation 1, that instead of a oxygen, it has a sulfur. Then there are other modifications that differ a little bit more from the structure of the deoxynucleotide. Some are really different. Some produce very big differences. So a lot of, now we have 3 generations, and constantly we are having laboratories producing new and new analogs. So what we did is we produce EGS isosequential with the EGS that were active, and we determine their activity. So I anticipate you that the log nucleic acids were the analogs that really gave us a satisfaction because they work very well. So we had almost negative results with everything, but the log nucleic acids really work well. So this is an illustrative gel. The reaction that we are going to do is similar to before: a label mRNA, an EGS that could be RNA or analogs, and we look for the degradation. So you can see that when you use an RNA, the messenger RNA, the messenger RNA original has been degraded, and we produce this fragment. As expected, an active EGS RNA has to be active. But then when you see all the rest, only one gives you a result that is similar, and this is an EGS that includes log nucleic acids. Some of you may be wondering now, you have different analogs, why you have 2 log nucleic acids. And the reason that we have 2 is that you cannot make an analog with every residue being a log nucleic acid. You have to mix it with deoxynucleotides or ribonucleotides. So in our case, we had different mixes. In red, you have log nucleic acids residues. In black, deoxynucleotides. And we could show that the mix, the configuration of the mix is very important, because if you have this configuration, you have no activity. You have this configuration, you have activity. So we had to do a lot of experiments, and we concluded that this kind of format, what we call a gapmer, because you have log nucleic acids at the ends, and the internals are deoxynucleotides. Those are the ones that work. If you get any other configuration, it doesn't work. Now, that created another question. If you're going to make gapmers, how many in each flank? So that was another group of experiments that we were going to do. And before going into that, we tried to first demonstrate that it was worth continuing fine-tuning, because it has to be active in vivo. Experiments in vitro only are not good for this kind of projects. And now we could not make a recombinant clone. So what we did was we use another trick, an E. coli that is hyperpermeable because it has a problem in the cell wall. So it will take up the EGSs. So we culture the E. coli resistance to amikacin, and we throw the EGSs. So these 2 bars are controlled. That means that the cells after 20 hours grew very well in the presence of the antibiotic. But when we try the EGS in question, we see that we have a significant reduction in growth, not as much as we would like, very modest, but very significant. So again, in concept, LNA DNA gapmers can induce an RNase-P-mediated inhibition of fractional resistance. And then it was worth continuing trying to fine-tune and refine the configuration of these LNAs. And what we can see is that this configuration, the green one, and this is a kinetic of degradation by RNase-P, has an activity that is similar to the EGS that is made of RNA. And then also there is another configuration that it was as good. We did this experiment with several regions to give generality to the configuration, and some regions were better than others, but the purpose of the experiment was to show that the green configuration is the best one, and we keep getting the same thing. Green and purple, green and purple, green and purple are almost similar, or even in some cases better, to the natural RNA. So with that, I think we close the issue of the analogs. We really got one good analog. The EGS is stable and it's active. The next problem was how to put it inside the cells, and for that, we started to work with cell-penetrating peptides. This is a derivation of existing proteins in nature, HIV and Drosophila, they have proteins that when they are conjugated to biomolecules, they have the ability, the cell-penetrating peptide has the ability to move it inside, through the membrane inside the cells. So we created a compound that has the gapmer linked by this chemical compound to the cell-penetrating peptide. And we did an experiment in vitro to see if all these molecules now still has EGS activity. And as you can see, in a kinetic of cleavage of the messenger RNA, we can see the RNA-sp still can do it, even when this gapmer has all of these attached covalently to it. So that leads us to the final experiment that is now, if I take a strain that is resistant and I put this complicated molecule, will I get some inhibition of resistance? This is the experiment where you see this is a clinical strain. This is not a laboratory strain. It's a clinical strain that is resistant to amikacin. These are growth curves in the presence of different controls, and this is the growth curve in the presence of amikacin plus the inhibitor. And as you can see, you have two ways to see it. One way is, well, not much, you have some growth, which is true, but also you have a significant reduction, which is also true. So if I had to qualify, I say this is a modestly successful experiment. So we can conclude that we have the way to do these compounds to inhibit resistance, but we have to do much better. Now, to do much better, we can get compounds that are much more efficient in inhibition, or we could find another inhibitor and mix the inhibitors and they can act synergistically to produce complete inhibition. That is what we are doing at the present time. Now, one little piece of information is that since new analogs come to the market, we are trying to see if we can get an analog that gets a better antisense. Now, we tried an experiment with a locked nucleic acid of new generation that differs in the chemistry of the bridge. And what we observed is that we lost completely the activity. So that could be not good, like you could say, this is not going good because you lose the activity. But there is a good aspect to this, and is that small changes in the chemistry produce big changes in the activity. The first one, the first new generation that has the bridge different, lost the activity. However, we have two new ones that are coming and they are ready to be tried. It could be that we had drastic effects for the negative, but one of these days could have drastic effects for the positive. So in other words, we continue researching for cell-penetrating peptides that can make the penetration more efficient. We keep looking for more analogs that could give us more efficient activity. And we keep looking for new inhibitors that can be mixed with this inhibitor, so you don't need to have the perfect inhibitor. You need to have two that contribute. And how do you contribute? You contribute by inhibiting the activity. So whatever molecule that escapes the inhibition of expression, you inhibit the activity. How do we do it? We do it by regular enzymatic inhibition, or we do it by what we think it is protection of the substrate. So by doing these kind of things, we are going to produce a new inhibitor that could be very good per se, or it could be that synergistically acts with the antisense. And the good thing with these kind of inhibitors that inhibit the enzyme is that in the world of the beta-lactamases, those are already in the market. So probably all of us already, once in our lifetime, at least, we took one formulation that we say, I'm taking an antibiotic, but you really take the antibiotic plus the inhibitor. What happens is that the beta-lactamic antibiotics, they get 80% of the attention of research, 80% of the, 80 or more percent of the resources. So research in the world of the penicillins, first is older because it was the first antibiotic, and second, it gets the lion's share of the attention, the human resources, that the funds and things. But if we keep doing, we can replicate those results with other family of antibiotics, like the aminoglycosides that we are interested in. So how did we look for these compounds? We did brute force. We just look for inhibitors without thinking, going through all of the reagents that we have in the cabinet and try to see if we find something that inhibits. We could ask the computer to do it, and these experiments were really good at producing inhibitors. The kinetics, the enzymatic kinetics of inhibition works like a charm, like a book, but they are not good in vivo. The bacteria normally can metabolize these products, and then you get no inhibition in vivo. So I'm not going to talk about this. And the other aspect is combinatorial libraries. Just get mixes of close to 1 million compounds and try to see if you get activity, and then they convolute that mix and try to find the good compound. So in a few words, the brute force that we didn't have any confidence at the beginning gave us very much a lot of satisfactions. So when we put zinc chloride, nothing simpler than zinc chloride, we already found that when we measure activity of the enzyme, we could inhibit the enzyme. And 15 micromolar of zinc chloride, already 50% inhibition. So really low concentrations gave us a lot of inhibition, very surprising. Different aminoglycosides all across the board were not acetylated in the presence of zinc chloride. So that was very exciting. We tried to do the experiments in vivo, and the experiments in vivo didn't work. But after like 2 or 3 years of thinking, how can we make something that works so well in vitro work in vivo? One day reading a paper, I realized that you could use an ionophore. And if you use an ionophore, then the complex will, zinc ionophore will penetrate the cells with very high efficiency. And that is when we realized that we could have something in there. In here, I'm showing you an experiment in which we have an E. coli resistance to amikacin, where we put different concentrations of zinc chloride just to show that we are not being intoxicating cells with zinc chloride. But when we put amikacin, we can put 1 or 2 millimolar zinc chloride, nothing happens. And that was because the zinc was staying outside. We was not going inside. But when we put an ionophore with 2 micromolar, we could completely inhibit work. So we could go thousands of times lower in concentration, and suddenly this was working. So this was tremendously exciting. As soon as we published, another group published a paper and confirming our results. And now this is one of those cases that you have to be careful with what you wish, because now we are having a lot of competition with this. There are like 3 or 4 groups working on this. But we are still happy that we were the leaders, we were the pioneers that found this result. This is another experiment showing that this is a Klebsiella that killed the neonate of meningitis because it didn't respond to amikacin. And then when you see this, you get, oh, why didn't we move faster? Because you can see with simpiridium, this Klebsiella dies completely in the presence of physiological concentrations of amikacin. This is a baby that was given the amikacin, and the Klebsiella grew and killed him. And this could have been a solution. Of course, you cannot, even if the results are good in the lab, you cannot go and give it to them. But if you go through the process one day, this could be the inhibitor, similar to the ones that we use with beta-lactamases. And then we wanted to show that it was not pyrithione, it was ionophore. So we look for other ionophores. And then we look for the families of the 8-hydroxyquinolines, and cleoquinol did the trick. So cleoquinol is a compound that is very good to be active for us because it's being researched as an anti-cancer for neurodegenerative diseases. So all the cytotoxicity, a lot of the different problems that you have in drug design are being resolved by all of the people that are working with this compound. And cleoquinol, this is a clinical strain as Acinetobacter baumannii. And when we added cleoquinol with zinc, we also got good results. The strain dies as if it was susceptible, despite the fact that it's resistant. So this was a very exciting development. We don't know the mechanism. We suspect that it's similar to something similar to what I'm showing here. The metal complex is somehow the antibiotic, and the antibiotic is disguised and it's not a substrate of the enzyme. Here you see, we could see this being researched with copper. I didn't mention, but copper and silver also do similar things as the ones that I spoke by with respect to zinc. So in conclusion, zinc is an inhibitor complex with ionophores. It can be used in concentrations reasonable for drug design. And they are active against at least 3 gram-negatives that are being problematic in the hospitals. So this is something I think has a lot of future, a lot of room for development, and it's a very exciting subject of work in the laboratory. And I'm going to conclude to quickly show our work on combinatorial libraries. So a few words about how it works. So we have a chemical group that is the scaffold and locations that can be replaced. So if you replace one location, then suddenly you can have a hydrogen of methyl, you have 2 compounds. But if you can replace the other, then you have 2 times 2, 4, times 2, 8, times 2, 16. And if you can keep adding possibilities of replacing the combinations, you can get to 800,000 or 1 million compounds. So what we did is we joined with the Torrey Pines Molecular Study Institute that had a lot of libraries, many scaffolds. So every bar that you see here means one reaction in which we add 1 microliter of a mix of compounds in the enzymatic reaction to see if there is inhibition. So when you see here this bar that says 35% inhibition, it means that in that little amount, we are putting 800,000 compounds. And you have 30% inhibition, and the inhibitor is 1 or 2, it must be very powerful because very few molecules of one of them are present. So we did this ranking. So some scaffolds didn't inhibit, but many did a good inhibition. So some compounds like the pyrrolidine, guanidine is one to be analyzed in the future. This benzodiazepine replace also seems to be good. I don't know if we are going to do analysis on this, but the one that we are doing analysis and are giving us good satisfactions is the pyrrolidine pentamine scaffold that has 4 locations for replacements. So the replacement, that is just to show the long list of replacements. And the number of total compounds is close to 750,000. And in here, I have to tell you, we have 1 mix that has all compounds, but then we have 120 mixes in which one of the R groups is constant. So if we have 26 possible replacements in R1, we have 26 mixes in which the R1 is constant and these are variable. And that goes for all of them. That makes 120 mixes. So what we did was do the reactions with every mix and see how the inhibition works. So for example, in this ranking, we have one compound that is the one that inhibits the most. And you know what the R1 is because it's constant. So in an ideal world, that would be the end of it. You said R1 is a phenyl, R2 is a phenyl, R3 is a phenyl, and this, and that's it, I have it. We don't live in an ideal world, so we have to do more analysis because this is not perfect. So what we do is we select a good number of those who are good perspectives, and we do dose response. When you do dose response, you see that some of those who look good are not really that good when you start reducing the dose that you use for inhibition. But some of them keep being good. But now you have less compounds. You have like 40 or 50 compounds that are left that are the finalists that you are going to analyze individually. And when you do the individual analysis of those few compounds, immediately you can see if you have one or more that are good, and those are the ones that you pursue. And if you see here in our ranking, compound 206 had an IC50 that's 2.1 micromolar, which is really good. We would like to have nanomolar, but we are close to nanomolar. So this compound is a very good inhibitor of the enzyme. And as you can see, it was not the one that would have been theoretically predicted. Yes in here, but not in here. You have to move a little bit to the left. So you need to go through that process, but then you isolate a good inhibitor. These are the R groups that are the winners for our 206. This, I'm not going to go in details into the kinetics, but it shows that you can get good enzymatic kinetics. You know that this inhibition is uncompetitive with respect to acetyl, and it's a mixed inhibition. And then you do checkerboard assays in which you take the 90 well plates and you start growing cultures at different concentrations of the antibiotic and different concentrations of the inhibitor. And red means good level of inhibition. You can see that there are combinations that are low in concentration and high in inhibition. So again, I'm not going to go into all of the math of all of this, but this just as an illustration. In experiments in vivo, we could see, in vivo or in cellular, because it's in cells growing, low concentrations of the inhibitor could inhibit expression. And then to conclude, we did an animal model of infection that luckily the Galleria melonella, that is these worms, is very well accepted as a good model. And we treat these models with Acinetobacter baumannii and Klebsiella, two clinical strains, and we try to treat the disease with amikacin and compound 206. And you can see that when you add amikacin, the cells survive, indicating that now we went beyond a cell in culture. Now we went into, again, not the best animal model, but an animal model that is accepted. So this kind of combinatorial library has a lot of potential, a lot of future, and compound 206 proved to be not cytotoxic to eukaryotic cells. So in consequence, we think that this is also a good way to go, and this is what we are doing now. We are going to try to refine 206, changing further these R groups to do structure-activity relations and get a yet more powerful compound. And we are doing that in conjunction with the group, the Torrey Pines, as I mentioned, and we now we included the biophysicists that are doing docking in a professional way with very powerful computers. So we are looking for a compound that is still stronger than compound 206. This is something that is ongoing at this moment. So the combinatorial libraries are a good way to get inhibitors. So you can see we have 3 lines that are being reasonably successful, and we don't need to have the ideal inhibitor. We need to have 1 or 2 that in combination could produce the results that we desire. So I will conclude by thanking the group of people that worked, that did the experiments, and here groups, some people that already are out, and actually one of them is going to be the next speaker that did the whole process, and now he has his own company. He's a founder of a company. So this is a great happiness for us to have seen that this student did so good in our lab, went out, get the PhD, the postdoc, went to Genentech, and now he has his own company. I'm not going to steal his thunder. And in blue, the groups that collaborate with us, the Torrey Pines Institute, now today, Florida International University, the combinatorial expert, mathematician, chemical synthesis, the biophysicist, and Dr. Ramirez group and Dr. Jimenez group. That Dr. Jimenez does all the cytotoxicity for us. We couldn't do that without her. And Dr. Ramirez, that she has been collaborating with me when she was I share her direction as a graduate student, as a postdoc, and now she's a very successful faculty, but we keep collaborating. And thank you to the agencies that gave us the funding. So with this, I'm going to conclude, and I'm going to be available for questions, if any.
Thank you, Dr. Tomasky. If you have any questions, please either raise your hand and unmute yourself or put the questions in the chat, please. I have a question, actually. I hope I don't get in trouble with the questions. So, when you were talking about the permeability issues of the EJS technology, is any of the developments that have happened associated with the COVID-19 vaccines can help with that? As some of that technology is to increase permeability of the RNA.
And also, permeability and also stability, because mRNA that was. I think, we didn't do anything, but we are thinking permeability not so much, because with COVID-19 you are talking eukaryotic cells, and the cell wall in bacteria is very different. So permeability, maybe nothing is translatable. Now the stabilization of mRNA could be. On the other hand, it looks like we already found the compound that is stable, that does exactly the same as mRNA, because the gapmers had the same kinetics and the same final activity. So now what we are looking for is something that is even better. So now to work with RNA may not give us advantage, because now we have something that works like mRNA. But that is something that, we are thinking and we are paying attention, because there may be something that could be transferred to us. But I would say that as of now, permeability doesn't seem to be, it's a different thing. And the stability of the mRNA maybe is a little bit too late for us, because we found the solution, a different solution, but the solution. So now we want to go beyond that. So who can see this transcript?
Any other questions for Dr. Tomasky?
I see something. Okay, if no question, I think we can invite people to go to the posters.
Yes, so I'm posting in the chat. I hope everybody can see it. The direct link to join Slack if you haven't done it before. We have wonderful posters, so I invite you to go and check on the posters. And then on each channel, you should have a Zoom link that the students had set up, so you can go and chat with them and ask all the questions that you have about their work. And our next talk will be here in this Zoom link at 10:40, right?
Wait, 10:40?
The next talk is 10:40, yes. Okay. So the posters are 10 to 10:30, and then 10:40, we have the next talk here, Dr. Lin. Thank you.
Thank you.
I have a question for the Slack, if anyone's still there that can answer. So I think, oh, I see that now. Rihanna Campos does have a Zoom link. Maybe I'm looking at an old spreadsheet. Is this one in email or?
Yeah, I sent it out last night with her updated Zoom link. And
oh, okay, thank you. I think I thought I had the most recent one, but I guess I did not. Thank you.
so I'll have it here if you can see it here too.
Hi, Marcelo.
So I think I can start introducing our next speaker. It's a very big satisfaction to introduce this speaker because he was a master student at Cal State and he is a paradigm of success. So that's how we want every student to do. So he graduated from UC Davis as an undergrad, then he worked in industry, he worked in public health and in life technologies before deciding to do the master, to pursue the master degree, which he did in my lab working. And as you could see, I have him in the acknowledgements of my talk. So after that, he went into a PhD program at the University of Michigan, where he did virology, in particular virus replication. And after that, he did a postdoc in Genentech, working on enteric neurons. And after that, he became a scientist in a biotech company, Twist Biosciences. And then he decided that it was time to fly higher. And he is now a co-founder of a startup biotech. And I cannot wait for the day when he goes public and we see the complete success. He's already a success, but that day is going to be a good day. So his company is Bristle, and you can ask him after his seminar if you're curious. But now it's his show, so I'm going to pass the podium to him.
Thanks, Marcelo. All right. Let me share this. Oops. Hold on 1 second. Typical technical difficulty. All right. Can everyone see this? Good? All right.
Yeah,
thanks,
we
Marcelo,
can see
for
it.
that great intro. Special thanks to Marcelo, not only for inviting me to talk, but also for allowing me to do my master's in his lab. Something he didn't say was that I was a very poor undergrad student, and he actually took a big risk on me. I owe a lot of what I've accomplished so far to Marcelo. Thank everyone for coming. I'm really excited to talk about my most recent kind of endeavor, which is this company called Bristle that I've co-founded. And the mission of this company is really to use the oral microbiome to improve health outcomes. Right now, we're really focused on oral health. And there's a number of reasons for that. And the first is that oral disease is incredibly prevalent. It's the #1 most prevalent disease in the world since 1990, and this includes both cavities and gum disease. I'm sure you know of somebody who's gone to the dentist and who's had to have their cavities filled or a deep clean for their gum disease. We spend about $50 billion every year out of pocket just for dental care. This is quite outstanding, especially compared to medical costs, and it's mostly because the systemic kind of health, your medical insurance doesn't cover oral health problems, and so most people have separate dental insurance, and we spend a lot on that. There's also 2 million visits to the emergency room every year just for oral disease. So this is typically because of emergency root canals or teeth being lost because of loose teeth from gum disease. Finally, about half of all adults over 30 have some form of gum disease. This is striking, and most people actually don't get it treated. Most people don't visit the dentist every year, or they don't have convenience to go and see a dentist. And these diseases are entirely preventable. So cavities and gum disease are actually bacterial infections. Both of these are there is some genetic component, but it's mostly because of your environment. It's mostly because of bacteria that live in your mouth. And we've known this since the 1700s. So this is just a letter from Anton van Leeuwenhoek, who wrote this letter, and he was looking under a microscope at a swab or a sample of someone's cavity. And then at the time, there was this hypothesis that worms were responsible for cavities because of the holes that they made in teeth. And this was kind of, there was an analogy to earthworms that were digging in the ground and made holes in the ground. And so at the time, Anton, he took a sample and put it under a microscope, and he saw what he thought were worms, but really they were lactobacilli, because he said that they were very similar to these other samples that he had seen from cheese. And it turned out that those were lactobacilli. So just to elaborate a little more on this, you're probably familiar, but cavities are really caused by these bacteria that create acid. And so what they do is they feed on the sugar in your mouth. And through fermentation, they create acid, and this damages the enamel on your teeth. And over time, this accumulates, and you get a hole in your tooth that can no longer be repaired by the natural process of fixing your enamel. And at that time, then you have to go in and you have to get a filling. But this process is reversible. So if you catch it early enough, it turns out that your enamel can repair. It's really because of the bacteria in your mouth. If they accumulate, they create this irreversible hole in your tooth. And the same also applies for gum disease. So what I'm showing you here is a picture, a cross-section of a tooth. This is a healthy tooth. At the bottom are the roots of the teeth, and at the top is the cap, and there's two different sides of your gums. Typically, you have homeostasis. So the bacteria in your mouth actually live at your gum line. They live all over your mouth. And typically they are in symbiosis. So they maintain homeostasis. There's a constant flux of inflammation that keeps your gums healthy. And this makes it so your teeth kind of sit in place properly and you can eat your food. But what can happen is in a state of dysbiosis, you can start to accumulate these bacteria that can invade the gum line and cause increased inflammation. And so this is an unhealthy tooth. So on the left side of that tooth, you have gingivitis. Gingivitis is the earliest stage of gum disease. It's just an inflammatory process where you have a buildup of plaque at your gum line, and they start to invade down into the roots of the teeth. And as it progresses, you can end up with an irreversible condition called periodontitis. Okay, and again, this is very similar to cavities in which they actually bore a hole in the root of the in the bone of the teeth, and at this point, it's no longer reversible, and you have to get surgery or some other process to fix periodontitis. And this only happens in conditions where you have dysbiosis. So there are bacteria here that are specific that cause periodontitis, but it's not just one species. So something that's come out more recently in a lot of microbiome studies is it kind of questions Cook's postulates, where a single pathogen is responsible for disease, because we find that it's actually a community that causes disease, because it's the interplay between some inflammatory pathogens and other keystone pathogens that can seed disease. And so just to reiterate, the left side of the tooth, gingivitis, is reversible because you don't have bone loss yet. So you can get rid of the pathogens that can kind of further inflame the region, and it's reversible. But once you get to the periodontitis on the right, it's no longer fixable. All right. So bristle, what we're trying to do is kind of change the existing standard of care, right? So what happens right now when you go to the dentist is they use these observational tools like an X-ray, or they probe you, and if you've sat in the dental chair, you know that they poke every gum, every tooth, and they're really measuring for a pocket, and they're seeing how much you bleed, because a sign of bleeding means that disease has progressed so far that they need to provide some intervention that's probably surgical and probably very expensive. Something else that's interesting in the current standard of care is that it's not objective. So if you go to one provider and they recommend a certain type of deep clean because of how deep your pockets are, there may be another provider who recommends something completely different. And this makes it hard really to take control of your oral health. And so what Bristle is trying to do is to modify this. We want to move to a more proactive management, because we think that, or we know that oral disease is entirely preventable. And the way we're going to do this is by not measuring the symptoms of disease, but by measuring the bacteria that are in your mouth that can cause disease. And so this is really going further upstream from the current standard of care. And this enables us to use a lot of tools that already exist to prevent the onset of disease. So there's things like high fluoride toothpaste or activated chlorine dioxide, and xylitol that can limit the burden of certain disease-causing pathogens. We think that by sequencing the oral microbiome, we can move toward a more personalized regimen that is not subjective and entirely data-driven and objective. Okay, so just a real quick pitch. Bristle, what we do is it takes you 5 minutes. You can order a kit from your just from the seat of your chair right now. You go to bristlehealth.com, and it's pretty quick. You just do a survey. We send you a box. The box is a sample, a saliva collection kit. So our entire platform is built on just using saliva. So it's not invasive at all and relatively quick. And from there, you send it back to the lab and we sequence your oral microbiome. And we measure kind of a number of factors that I'll get into, and we give you your risk for gum disease, cavities, other oral health conditions, eventually also systemic health conditions. I'll also get into that. And you can also track your status over time. So let's say a provider asks you to change the toothpaste that you're using to some prescription grade toothpaste. If you want to track whether or not it's working, we have a platform for that. Okay. All right, so jumping into the science. So how do we do this? All right, so what we do is we take your saliva, okay? And from your saliva, we purify out the DNA and the RNA. We do an upfront kind of, we call it a host depletion step. And the reason we have to do this is because saliva is actually, somewhere between 60 to 95% human DNA. And I think I may have mentioned this, but your oral microbiome is entirely driven by your environment, and we're not that interested in your host genetics. It may be something that we tie in later, but right now we don't look at host DNA, so it's not like 23andMe. Instead, we look just at the bacteria, fungi, and viruses that live in your mouth. And so we do this upfront host depletion step, where we try to remove as much of the human DNA as possible. And then we do sequencing library prep. And here we're using something called Shaka metagenomics that I will touch on later, but essentially it comprehensively looks at all the DNA or all the RNA in the sample. So we don't bias ourselves to looking at targeted sequencing or amplicon sequencing, because we want extremely high resolution. And this means we want to go all the way down and look at like the strain levels of each of the species that live in your mouth. All right, and then we do some sequencing. And then, after sequencing, we run it through our analysis pipeline. And this pipeline really involves first the dehosting step. So again, we remove all the human DNA, we do some quality control, and then we do a mapping step. So the mapping is really key here. So we map to reference genomes for all of the microbes that have been sequenced to date, including the ones that are in our own proprietary database. And this is really key because so we're limited by reference sequences, which is one issue, but because we're using Shaka metagenomics, we can actually expand the database, and we can grow our own internal database of all the oral microbiome kind of strains of bacteria, fungi, and viruses that we ever find. And that allows us to do some really deep kind of learning into what's in everyone's oral microbiome. Okay, so I mentioned that we use Shaka metagenomics, and there's a few reasons for this, and I'm just going to go through kind of what has been done historically from right to left. So long ago, when people did dental microbiology, they would take a swab and they would just put it on a plate, right, and see what grows. And this is not quantitative, right? You're just looking at presence or absence. You're seeing if there's an infection of some kind, because you can grow these up and then you can put them on biochemical assays and use a Bergey's manual that you all may use still. And this requires culturing, takes a long time, not high throughput. And one step above that is to use qPCR. And so this is very targeted. And there are a few companies that do this kind of testing. And really, they only look at somewhere between 10 to 20 different microbes. This doesn't give you any resolution. It's really more of a presence-absence test, because having an exact quantitation by qPCR is actually very challenging. And then one step above that, beyond using predetermined targets, is do 16S sequencing. You're probably all familiar with this by now, but 16S sequencing uses a targeted conserved region of the bacterial genome, and you can sequence this out, and you can infer what species existed because 16S is, there are SNPs in 16S that you can use to map to different bacterial genuses and sometimes species. And the issue here is the resolution is not very high. So just to give a COVID example, if we sequenced out certain regions of COVID, we would not be able to determine if it was Delta or Omicron, right? Same for qPCR, you can't really tell what variant it is unless you sequence the whole thing. And so shotgun metagenomics really allows us to do that. So we use whole genome sequencing. One limitation, of course, is the human DNA in there. But it allows us to get very high resolution. We can go all the way down to the strain level, and we can identify not only bacteria, but also fungi and viruses. All right, so shotgun metagenomic sequencing, the output looks something kind of like this. So what I'm showing is just a graph. On the x-axis are different patient samples, and on the y-axis are strain level relative abundances, where each of the bars contributes to 100%. Okay, so if your microbiome is a pie, then each of these is a tiny slice of the pie. And you can see that each of these patients has a very different oral microbiome. So each of the gray bars, if you just take a look, their distributions are very different, right? And so this is really complicated. Like there's a lot of information here. And so the goal of Bristol really is to take this and to turn it into something that's very easy for people to consume. And we turn this into kind of a score. And so we have an internal way to do this. And this is kind of the information that we give you. We actually give you both. We give you your raw oral microbiome. You can play around with it. You can look at all the data. And then we also boil it down to a score for you. And I'll explain a little bit about how we do that. Okay, so we had built this platform, and we built this platform off of a few initial samples and some publicly available data sets. And so what we wanted to do is really prove that it works. Okay, and the way we did this was we collaborated with a dental school here up in the Bay Area in San Francisco called University of Pacific. And we recruited participants ages 18 to 70 to donate their saliva. Okay, so all of these patients, they come to the dental clinic. It's a free clinic because it's a teaching clinic. And these participants, they donate their saliva. And then immediately after that, all these patients, they undergo a dental exam. Okay, and so that means that we know at the time of collection, did somebody have gum disease? Did somebody have cavities? And other oral diseases. So they also check for oral cancer and bad breath and a number of other things. We sequence the saliva, like I mentioned. And then most importantly, at the end of this, we actually can back correlate all of the data. So we match all the sequencing data to electronic health records from these participants. And the reason this is so important is because this kind of metadata really doesn't exist anywhere. And so we're able to derive a lot of things, not just about their oral health, but also about systemic health, about their lifestyle, about about their systemic health conditions. Do they have diabetes, cardiovascular risk, and a number of other very interesting pieces of information. Okay, so the first thing we did was to see, did our assay work in this condition? Okay, and so I told you a little bit about cavities causing bacteria. And one of them that has been known canonically is called Streptococcus mutans. And this is very common in children. And children with strep mutans, pretty much 100% of the time, if you have Streptococcus mutans in your mouth, you will succumb to cavities of some kind. And it's because strep mutans is really good at growing in the presence of acid, and it also creates acid. So it's a fast-growing organism that can form colonies on your teeth if you don't remove them well, or if you don't have great oral hygiene, like most children don't. So here on the y-axis, I'm just showing the strep mutans relative abundance, and on the x are categories from electronic health records. And so in this study, what they had was 4 different categories of cavities risk. And the way that this is measured is there is a survey on certain things called Cambra, and it also involves an ATP test that kind of measures the bacterial burden in your mouth. And it's that paired with, how much fluoride are you using? What is your diet like? I think there's a series of probably between 10 to 15 questions, and that contributes to this. An extreme risk is very high. It means basically you have existing decay that is untreated. High means you have you had decay before, and you might not now, but it still required treatment. Moderate and low are other lower categories. And you can see that there's this kind of trend where strep mutans is much higher in people with extreme risk for cavities. And so this is a great validation of our system that, maybe this is working. Like it looks like we can pick up some cavities causing species, especially in people who have extreme risk of cavities. Okay, but Shaka metagenomic sequencing data is actually really complicated. Right? It's not just Streptococcus mutans. We're looking at a community. We're looking at hundreds of species. And that actually looks something like this. So here in this graph on the y-axis are different bacterial species. And on the x-axis is this measure that we call importance. So basically we run the data through a machine learning pipeline, and it outputs something that looks like this. And it's pretty much nonsensical. Unless you feed it through a computer that gives you an algorithm, and it's just a black box. Okay. And so as much as I can mention, we have an algorithm that takes all of this information and it spits out a cavities risk. So we call them the bristle cavities risk score and a bristle gum disease risk score, a halitosis risk score, a number of other things. And it takes into consideration, what are the good microbes in your mouth that can be preventing the bad microbes from growing out? Does your community look more like somebody who has cavities, or does it look like somebody who doesn't have cavities? And the same for gum disease and a number of other factors. And then we come up with a score that goes from 0 to 100. Okay, with 100 being the highest risk and 0 being the lowest risk. Okay, and so we applied this algorithm to a much larger data set. Okay, so we collected more samples from the clinic. We blinded our assay, and we threw the algorithm at all the data. And so what I'm showing you here is just on the y-axis bristle cavities risk score. On the x-axis is a diagnosis. Okay, so all of these samples have a matched diagnosis at the time of sample collection. And really, we're just asking a question: did this person have cavities or did they not? And what does their oral microbiome score look like? Okay, and so these are just box plots. And if you just take a look at this line is, I think here is the median. And you can see that in people who have cavities, if they have a yes, that their bristle caries risk score is much higher. It's more than 2-fold higher than people who don't have cavities. Okay, and so far it seems like this is working. And we do a little bit of statistics, and we come up with this thing called an area under the curve. And this is kind of a diagnostic score. So this is you may have heard of this now because of a lot of COVID testing. But basically an AUC of 100 is perfect. There is no test that's perfect, but 100 would be perfect, which means it's 100% sensitive and 100% specific, which means there's never a false positive and there's never a false negative. So the goal really is to reach something above 90. Above 90 would be an excellent AUC. Above 80 is an acceptable, at least in my perspective. And so far, it actually looks like, we can measure cavities with pretty good accuracy with never even seeing the patient. This is just using oral microbiome. If we also tack on survey information and familial risk, we can increase this AUC significantly. And we did the same thing for gum disease. And so again, the graph on the left shows the gum disease risk score, and on the X, it's their diagnosis at the time of sample collection. Did they have gum disease or did they not? And here, we're only looking at irreversible gum disease. So we don't look at gingivitis for a number of reasons, mostly because 75% of people have gingivitis, basically. Almost everybody has some form of inflammation in their gums. And it's usually pretty healthy. It's good to have inflammation, like I explained before, but at some point it becomes irreversible because you've had bone damage. And here we're just looking at the people who've had this kind of very severe damage that can't be reversed. And it looks like our algorithm works really well, and that we can pick up people who have gum disease versus those who don't. And the AUC for periodontitis, which is irreversible gum disease, is 88. And so we're approaching kind of, very good area. We also looked at some other very interesting, this is more data that kind of validates that the assay works. And so what we're showing here is on the y-axis is the gum disease risk score, and on the x is the number of times that somebody flosses every week. Okay. And so what I didn't tell you is that the microbes that cause gum disease are typically anaerobic, so they can't grow in the presence of oxygen. And the purpose of flossing is really to get beneath the gum line to remove biofilm and to aerate the region. Okay, so as you put floss at your gum line, you're introducing oxygen, and this should presumably, if you're doing it properly, it should remove anaerobic pathogens. And so we actually see that in the data. So it turns out if you don't floss at all, your gum disease risk score is pretty high. And if you floss even just once a week, your risk score decreases dramatically. And as you increase your flossing, if you get to 8 times or more, you may be improving your oral health score. But there is a major caveat here. It's that people who floss 8 times or more already care more about their oral health than someone who never flosses at all. So there's a lot of confounding factors here because all of human biology is basically edge cases. Everybody does something different in their mouth. They all have different diets. So there's a lot more to tease apart here, but so far this data looks quite promising. Okay, and so for the last few minutes, I just thought I would talk about a little bit about where I think that bristle is headed. So right now we have this great assay that can, kind of predict your risk for gum disease and cavities. But there's a lot more we can do with oral microbiome data. Okay, and so there was this paper in 2019, and it showed that this pathogen called Porphyromonas gingivalis was actually in the brains of people with Alzheimer's disease. Okay, and this is really striking because there was a hypothesis at the time that there was a gut-brain connection. So some of the microbes in your gut may be responsible for causing neurological disorders. And the same had been postulated for the mouth, but no one had really looked. And so in this paper, they really looked. And so Porphyromonas gingivalis, as you might derive from the name, is a gingival pathogen. So it causes gingivitis. And if you have a lot of it, it's actually responsible for periodontitis as well. So what they did was they here they took the brains of people with Alzheimer's and they measured RGPB is a gingipane. So a gingipane is a protein, it's a virulence factor protease that is derived from P. gingivalis. Okay, and the red triangles here are people with Alzheimer's, and the blue circles are the people who are healthy. And so, as you can clearly see, there's a trend where you have more kind of RGPB in the people who have Alzheimer's versus those who don't. And they also did some immunofluorescence just to show that this gingipane actually somehow ends up in their brain, and they co-localize with these tau filaments and these beta amyloid plaques that have been also hypothesized to be responsible for neurological decline. And so since then, about 3 companies have popped up trying to target these gingipanes and target these pathogens to reduce their burden, and to see if we can approach Alzheimer's disease as really an oral health issue, and not just looking at the brain. Okay, but it's not just Alzheimer's. There's actually a lot of different diseases. So your mouth is seeded with all these microbes, and your oral microbiome is really complicated. And everything that you eat ends up everywhere else in your body. So like I mentioned, it has a correlation with Alzheimer's disease. There are certain bacteria like Fusobacterium nucleatum that have been found in plaque in people's arteries that have had heart attacks. A lot of these bacteria also end up in the gut, where they can kind of increase inflammation in the gut, and also have been implicated in advanced colorectal cancer. And they may also be responsible for asthma, adverse pregnancy outcomes, and diabetes. And just to show a little bit about what we've done so far is we've asked people who have taken our tests, have they been diagnosed with diabetes? And we look at their oral microbiome. Here I'm only showing you on the y-axis the cavities risk score, because you'd expect that people with cavities, well, actually people with diabetes may have an increased risk for cavities because their saliva would have an increased sugar level. And it turns out that may be the case. We can actually find that if you've been diagnosed with diabetes, your score may be a little bit higher for cavities risk. What I'm not showing you here is the species that have been, that we've identified, the strains that we've identified as being kind of differentially abundant in the people who have diabetes versus those who don't. And that also is some very interesting information. So just to conclude, oral health, big problem. People don't really pay attention to it because it's not tied to your regular healthcare. And Bristol uses the oral microbiome to look at your oral health. And we can do a lot of interesting things to predict kind of where we think your health is headed and how you can mitigate that by taking one of our tests. And we're always looking to collaborate. So we love data, we love science, and if you have any collaborations that you're looking for, we're always willing to talk. So you can send me an email. And if you're interested in participating in Bristol, feel free to go to our website, bristolhealth.com, and just pick up a kit. Thanks.
Thank you, David. Really, really interesting talk. I think there are some questions in the chat.
Okay, I can take a look. I'm gonna stop share real quick so I can see. Otherwise, I can't see the chat, unfortunately.
I can read it to you if you need.
Okay, yeah, sure.
So Ingrid is asking if there is a difference in the oral microbiome between young and elderly people.
Yes, absolutely. So this is a great question. I didn't show the data, but the risk for gum disease increases significantly as you age. And we do find that there are some pretty interesting shifts that happen from people who are younger to people who are older. As you get older, the periodontal pathogens tend to show up a lot more. Why this happens, we're not entirely sure. It could be because of, your immune system is not as strong as it used to be. So there is a steady decline in your immunity. And this allows these pathogens to kind of, the homeostasis levels change because these pathogens can now outcompete your immune system. That is something that we're actively looking at. It's also, it's not just age, it's also gender, and it's also race. Yeah.
I have a follow-up with that then. In your saliva test, can you pick up some immune signals from the person actually to kind of correlate that factor into your risk calculation?
Yeah, that is a great question, and that is something we're actively working on. So what we want to do is metatranscriptomics. So we want to look at the transcript levels of not only the bacteria and the fungi and the viruses, but also of the immune system, right? Because a lot of these factors have been implicated in the progression of oral disease and maybe also systemic disease. And so, yes, that is something we're looking at, and our saliva test can do.
Great. There is a question from Melvin. Are you planning to come up with a specific antibiotic treatment against specific bacteria found in the mouth, like precision medicine approach?
Yes, absolutely. That is the goal for personalized medicine. Of course, you're all familiar. It's quite challenging to do. But fortunately, actually, a lot of these companies that have popped up to tackle Alzheimer's, we've actually been in conversations with some of them to kind of retool their drug to address gum disease first, because gum disease is a very attractive option because a lot of people have gum disease. And it's easy to measure outcomes because either your gums improve or they don't. And we can also specifically measure the bacteria and if the levels have changed. And so, yes, we are trying to do that.
Right. Another question in the chat. So when collecting saliva samples, can you get different oral microbiome measurement score at different times of the day? If so, is there an optimal time to collect saliva to accurately score the microbiome?
Yeah, so we frequently get this question actually. And it turns out that your oral microbiome is relatively stable. When I say relatively, that means that, if you maintain the same regimen that you do every day, and you brush your teeth, you use your mouthwash, you floss, it's going to look the same whether or not you took it in the morning, took it at night, took it the next morning. So these big shifts really only happen when something changes or over longer timescales. So it's very similar to something like evolution, because that's kind of the process that's happening in your mouth, where over a long period of time, dust accumulates in the corner of your room, and all of a sudden it's really dirty, even though you think you cleaned it every day. But regarding the exact time of the day, there were some studies that show that there is a circadian rhythm to your oral microbiome. Like there's a flux of how often certain bacteria can outgrow others, and it kind of makes sense, right? Like in the middle of the night, you're breathing differently, your activity is different, you may not have eaten anything for a long time. So what we recommend is to take a test first thing in the morning, because this is when there has been zero intervention. And this provides us a snapshot of what it looks like when you haven't done anything. And so so far, that's what I can answer. We'd like to look at kind of circadian rhythms. and see if there's anything there. But those don't contribute to huge shifts in your oral microbiome. Yeah.
Another question just pop up. Can the use of mouthwash like Listerine damage your normal oral microbiota? If so, is it safe to use it all the time?
Yeah, good question. So we do recommend that people use mouthwash if they've been using it and they don't have any issues with their oral health. So I'm just going to go back to the kind of dirty room analogy. You clean your room every day, right? And you think it's clean, but over time there ends up these very small shifts, and all of a sudden, like, you look in the corner and there's a cobweb. It's like, how did that get there? I clean this room every day. And it's because of these small pockets. And so mouthwash is really similar to that. You're just kind of cleaning the room. You're actually reducing the levels of everything. It's a non-specific antimicrobial. And then, over the course of years and years, you keep on doing this, but there's a pocket in your mouth that, the mouthwash never hit. It doesn't really get beneath the gum lines, even if you swish it. Some people have, diluted it out and used it in a water pick to help you floss. But overall, we still recommend mouthwash because it does reduce the burden, the total levels of bacteria, but the community shift is really what's responsible for disease, and Listerine doesn't overall affect that.
Thank you, David.
I have a question. Hi,
Go
David.
ahead.
Great presentation. I was just wondering, you were talking about how there was a rise of that bacteria that thrived in acidic environments. Is something you do for your bristle test? Do you ask the people what kind of what their diet is like? Because I'm thinking, like, there's a lot of coffee drinkers out there, and that's very acidic. You have people that eat healthy, they're eating like acidic fruits and all that. So is that something you ask them to see if there's a correlation between those acidic bacteria and their diet?
Yeah, that's a great question. So I would say I'd love to ask those kinds of questions. I really like, getting into people's lifestyle and how that could affect their oral microbiome, because you're right, like drinking lots of coffee, if you just sip it throughout the day, the pH of your mouth is going to be much lower than someone who doesn't do that. And it's the same for soda or a number of other drinks, even foods. The problem that we have been running into with these kind of self-reported data points, especially for these lifestyle things, is that people don't know that they do something differently than other people. So for instance, we can ask you how often you drink coffee, but it's very difficult to ask you, how many times a day do you sip coffee more than X number of times per hour kind of thing, right? Because if you drink a cup of coffee, first thing in the morning, and then you don't touch it again, the pH of your mouth actually normalizes quite quickly, because saliva is really good at that. But like I said, is that human biology is all about edge cases. Like, everybody does something differently, and it's really hard to grasp at that. And so one of the things that we're trying to do with our platform is to get surveys that are very similar to 23andMe surveys. So these questions have to be yes or no. They can't be free form, and they have to be very granular. And so that's something we're actively working on.
Thank you.
Yeah, thanks for the question.
All right, thank you everybody for your very interesting questions, and thank you again to David, this awesome talk and great work. We have the next poster session from 11:30 to noon. So I invite you to go and see some other posters and chat with our students. And the last talk of the morning It's very promising and interesting, and it will be at 12:10 in this same link. Thank you, everybody. See you later. I hope you had time to see some of the posters and chat with the students. This is the last talk of the morning, and it's my great pleasure to introduce Dr. Lanning. He is an associate professor in the biology department at CSU Los Angeles. He obtained his PhD in cell molecular biology at the University of Michigan, and then went to the Van Andel Research Institute in Michigan too for postdoc work. And in 2014, he joined the CSU family. And me and my students are super excited about this talk because his work is in mitochondrial bioenergetics. And we also work in mitochondria, but in parasites. So we are really, really excited to know about his work on different aspects of mitochondrial metabolism associated with diseases. And without further ado, welcome, Nate.
Thanks, Veronica, for that introduction. And it's great to see all the students here. Of course, my monitor just shut off when I started. So yeah, I'm going to talk today a little bit about mitochondria and mammalian cells. So I'll get my screen share going on here. Somebody give me a verbal when you can see it. I can't see the meeting anymore. So just let me know when
we can see it.
All right, great. All right, so the title of my talk is Surveying the Mitochondrial Bioenergetic Landscape for New Therapeutic Targets. And before I get going, I just want to thank the organizers for inviting me here, and also really to encourage you all. It's great to see all the students here. It's wonderful. It's really hard to maintain some of that research and scientific momentum in this virtual environment. So it's great to see the participation, and frankly, I'm a little bit jealous. So kudos to all of you. And students, you can kind of take advantage of this too. Don't be afraid to tell your professors you need a little bit of extra credit to come to these things. But we'll start talking about mitochondrial bioenergetics and what does it mean to say that we're going to survey the mitochondrial bioenergetic landscape. Some fancy words there. We'll see if that's just fancy words or if it actually means anything. I'm not really decided yet. Before we get into the data, I do want to acknowledge those students of mine who have done the lion's share of this work. Jonathan and Ivan I have here listed first. I was actually going to try to fit 2 lectures into 1 lecture, and about half an hour ago, I realized that's a really bad idea. Jonathan and Ivan just finished a paper that got accepted this week on looking at a number of different mitochondrial genes in lung cancer, but we're not going to have time to talk about that today. An and Jesse, master's student and undergrad student in my lab, have done all of the work with all the signaling components that we'll talk about today. And then Carlos, a master's student, and Benjamin, an undergraduate, did all of the work on the redox sensing and reactive oxygen species. So those are the students in my lab who've done the work on the project that we'll talk about today. So let's talk briefly about mitochondrial biology. All of us know about mitochondrial biology. It's one of those prerequisite things that we talk about in our courses, in our undergraduate courses, in our intro undergraduate courses. We know that mitochondria are the powerhouse of the cell, right? That's what we always remember. If there's one cliche that we remember either from junior high or high school or intro college biology, it's that mitochondria are the powerhouse of the cell. And they're the powerhouse of the cell because when we're trying to do powerful things, that is, use energy to do work, we need mitochondria. So we have this Cal State Fullerton player right here dunking the basketball. He's using a bunch of energy in this to hopefully hang on the rim a little bit, show that he had a great dunk. He needs energy in his muscles to do that. And a lot of that energy is provided by mitochondria. So if we zoom in to his muscles and his tissues, you'll see lots of cells. Inside of those cells, one of the major organelles inside those cells are mitochondria. So this middle image right here, this green fluorescence, demonstrates how many mitochondria are packed into normal mammalian cells. So it looks like this cell is almost completely lit up with green, indicating that there's lots and lots of mitochondria in this cell. And that's really typical for mammalian cells. They're just packed with these things. Right here to the right of this image, we have a little schematic, a cartoon of a mitochondria as we usually see it in textbooks. But that's not what you see over here. Over here, you see this reticulated network of green. And that's typically what mitochondria look like in healthy mammalian cells. It's a reticulated network with lots of mitochondria connected to each other into these long tubular structures. It's much easier to draw them like this though in textbooks. So we draw them like this. And if we zoom into individual mitochondrion, we'll see lots and lots of stuff packed in there, including this. Here's a little depiction of the electron transport chain that we're probably all familiar with, and the reason for which mitochondria are called the power of the house of the cell, converting lots of chemical energy into a usable form, ATP. All right, so that's a brief overview, just a reminder of what mitochondria are. However, as many of us know, mitochondria aren't just the powerhouse of the cell. That's really a one-dimensional view of mitochondria, is to think that they simply take in nutrients and spit out ATP. They do lots more. They take in all different kinds of stimuli, all different kinds of signals. They interpret those signals inside the complex shape, the complex structures of mitochondria, and then on the other end, they give some sort of output. So a lot of mitochondrial biologists actually look at mitochondria and think of them as signaling organelles. And they think of them as signaling organelles because they're taking in some sort of signal, they're decoding that signal, they're responding to that signal internally inside the mitochondria, and then giving some sort of input to the cell. And if you look at all these different functional outputs right here, these are all related to some sort of cellular homeostasis, whether it's ATP production, maintaining the appropriate pH or redox status of the cell. Lots and lots of homeostatic cellular functions are maintained through this, what we call the signaling functions of mitochondria. And that's really what my lab is interested in, is understanding how these signals come into mitochondria, how the mitochondria interpret and then respond to those signals to maintain cellular homeostasis. And we want to do this by looking at all the different proteins and functions inside mitochondria, and we'll get to that in a minute. But one of the reasons we want to do that is because when you completely inhibit functions of mitochondria or alter mitochondrial functions, diseases can arise or diseases can be exacerbated. So if you completely inhibit mitochondrial functions, specific diseases such as neurodegenerative diseases and a category of rare diseases called mitochondrial diseases can result. And these are really a result often of inhibition of mitochondrial functions. On the other side, you can alter, either decrease mitochondrial functions or increase specific mitochondrial functions and support diseases like cancer or diabetes. Now, it is important to note that not all of these types of diseases arise directly because mitochondrial functions or proteins have been inhibited or altered. Oftentimes, a disease state will arise. It will impinge upon a mitochondrial function. That mitochondrial function will be inhibited or altered, and then that inhibition or alteration of that mitochondrial function will exacerbate the disease. So it's part of the etiology of the disease, it's part of the development of disease. It's not always part of the beginning of the disease itself. But we're interested in how these functions inside mitochondria are altered or inhibited, and then are associated with these different disease states. However, in order to look at all of these different inputs, all of these different outputs in the context of these diseases, we got to start somewhere. We can't study all of them at the same time. So the first thing that our lab did was to say, let's just study that basic function of mitochondria, wherein mitochondria take in nutrients, they process those nutrients, and they spit out ATP. We know those functions very well. We know the TCA cycle. We know how pyruvate is converted to acetyl-CoA. We know the electron transport chain. And those are really important for mitochondria responding to nutrients producing ATP. But we were also interested, are those the only proteins, the only structures inside mitochondria that are responsible for ATP production from nutrient signals? So it turns out there's somewhere between 1,500 to 2,000 proteins that directly localize to mitochondria in mammalian cells. So there's lots and lots of other candidate proteins that might be involved in responding to nutrient status inside mitochondria and producing ATP. There's about 60 different individual delineable functions in mammalian mitochondria as well. So it's possible that instead of just the TCA cycle or just the electron transport chain, there might be other functions that are really critically involved in ATP production in response to nutrients in mitochondria as well. So we want to start to pick apart these 1,500 to 2,000 proteins and see which ones contribute to the signaling outputs right here in response to these signaling inputs inside mitochondria. So today we're just going to talk about the nutrient signals and the ATP production responses though. To refresh our memory, there are a number of different types of nutrients. And here, when I say nutrients, I'm talking about carbon-based sources that can be converted into ATP through cellular respiration, through those basic programs in mitochondria. So we all know that glucose is involved in, we have this process of glycolysis where we have pyruvate, glucose converted into 2-pyruvate. The pyruvate enters the mitochondria and is converted to acetyl-CoA, hops into the TCA cycle, et cetera. What most of us sometimes forget is that most mammalian cells utilize glutamine at just the same levels and sometimes even higher levels than glucose to supply cellular respiration fuel. So glutamine can come into cells, be converted to glutamate, which then is converted to alpha-ketoglutarate and hops into the TCA cycle as well. Galactose can also participate in cellular respiration, and so can pyruvate. So these are all different carbon sources, carbohydrates that are floating around in the plasma of normal mammals, which come into cells and which are used as cellular respiration fuel to drive mitochondrial respiration. So we wanted to know what are all the different proteins inside mitochondria that are required to regulate ATP output in response to these different nutrients. Again, we know the different pieces and protein parts, the proteins involved in the TCA cycle, the proteins involved in the electron transport chain, but like we said, there's probably somewhere around at least 1,500 other proteins here in mitochondria. And we're wondering, are there any others that contribute to ATP production in response to these fuel sources in mitochondrial oxidation? So in order to address this question, we performed what we call the targeted RNAi screen to interrogate these mitochondrial associated ATP production from these different fuel sources. So RNAi, or RNA interference, is a technology that's been around for quite a while now, wherein we can feed small RNAs to cells, to mammalian cells. These small RNAs will inhibit the production of proteins specific to those RNAs. We call them siRNAs, or small interfering RNAs. So it's just a way to inhibit the specific protein expression of whatever protein we want. So our first pass at this was to take 1,000 different siRNAs. Each one of these 1,000 different siRNAs targeted a specific protein inside mitochondria. And we fed these to cells in the context of these 4 different mitochondrial oxidative fuel sources. So glucose, pyruvate, glutamine, and galactose. And 72 hours later, that's about the time it takes for proteins to degrade after siRNAs have been fed to them. We measured the relative amount of ATP per cell. So we're simply looking for how does total global cellular ATP levels change in response to suppressing the expression of these 1,000 different proteins through these siRNAs. And what we found is that across the 1, 2, 3, 4 different nutrients, right, the glucose, pyruvate, glutamine, or galactose nutrients, there were 11 proteins that were critical for maintaining ATP levels irrespective of those fuel sources. And those are listed here. There's the same group on top and the same group on bottom in all 4 quadrants here. That just means that when you suppress the expression of each of these proteins, you had the almost an identical effect in each of the 4 different fuel sources. So that was exciting to us. It seems that there's perhaps a core group of proteins that's important for maintaining ATP production, irrespective of nutrient status. And if you look here, AK4, this enzyme called AK4 was the protein that was right at the top of 3 of the fuel sources, and the second and the third fuel source. So that was exciting to us. It told us that maybe there was a protein that was particularly important for maintaining homeostatic ATP levels in response to various different fuel sources. So this AK4, the name of this protein is called adenylate kinase 4. AK4 is adenylate kinase 4. So that brings up the question, what are adenylate kinases? Well, adenylate kinases are important for maintaining intracellular adenosine ratios. If you remember your biochemistry, you'll remember that in most mammalian cells, there's a very strict ratio of adenosine, a very strict adenosine ratio maintained. So it's about 100 ATP to every 10 ADP to every 1 AMP. So a 100 to 10 to 1 ratio is maintained pretty strictly. And that's maintained by these adenylate kinases. So AK4 is one of these adenylate kinases. It happens to be in the mitochondrial matrix. There are 9 family members of these adenylate kinases. 3 of them, AK2, 3, and 4, are found in mitochondria. So these are the ones that were in our screen. And it turns out that suppressing the expression of AK4, if we take a look back, suppressing the expression of AK4 led to an increase in ATP levels. That's the y-axis on all of these graphs. Getting rid of that protein led to a significant and a repeatedly significant increase in ATP levels. So that was really interesting. Getting rid of something increases ATP levels. So our first thought was, well, this enzyme AK4 is typically going in this direction, consuming ATP and producing ADP. But eventually we did those experiments and found that that was not true. So there's some other reason whereby getting rid of this AK4 is leading to increased ATP expression. That screen that we did, that data that we just looked at, that was all in HeLa cells, a cervical carcinoma cell. We repeated the experiment in SF295 and U251. These are human brain cancer cells. We saw the same thing in all of the following slides. Gray is the control condition and blue is the AK4 loss or AK4 knockdown condition. You can see that whenever we get rid of AK4, we get a bump up in ATP levels. That's the y-axis again, irrespective of the type of nutrient that we're feeding these cells. So we did it in cervical carcinoma cells. We did it in these 2 brain cancer cells. We've done it in more than a dozen different cells, including many normal cells as well. These are all human cells that we've done these in. And we've seen the exact same trend all the time. There's always an increase in ATP whenever you knock down AK4. Doesn't matter what nutrient condition, doesn't matter what cell type, where it's from, whether it's a disease cell or whether it's a normal cell, an increase in ATP production. So we wondered, how is this happening? How is this occurring? So we thought we would look at the relative rates of cellular respiration. So there's a number of different ways whereby you can measure the rate of glycolysis or the rate of OXPHOS. So just glycolysis or mitochondrial oxidative phosphorylation. And we saw that there, again, it didn't matter what cell type we used. If you knock down AK4, there was a slight increase in glycolysis. There's always a slight increase in glycolysis, but there's almost a doubling in OXPHOS. So getting rid of AK4 somehow, some way dramatically increases OXPHOS activity. So the rate at which cells are consuming oxygen in mitochondrial respiration. So consistently enhances OXPHOS activity when we get rid of AK4. So that was interesting to us. How does that work? So we started to build a model to try to figure this out. So we know that when we suppress AK4 expression, we get a little bit of an increase in glycolysis, a big increase in OXPHOS. So there's a question mark here. How is that actually working? Our lab has also been very interested in how mitochondrial biology relates to cell survival. A lot of cells use mitochondria kind of as a sentinel for the health of a cell. And if the mitochondria get perturbed, that will either cause cell death, or if the mitochondria are very healthy, it will cause cells to stay alive. It will increase the viability of cells. So we were interested in this AK4 protein, getting rid of it, and how that relates to viability in response to nutrient deprivation. So you can take specific nutrients away from cells and see how that impacts cell viability or cell survival when you take those nutrients away. So you can see in these two different control conditions with these two different cell lines, again, you have the control conditions, 100% viability, and then relatively, if you take away glutamine or glucose, you decrease viability to some specific extent. However, if you do those experiments in the context of suppressing AK4 expression, you rescue viability all the way up to 100% in many cases, and significantly in pretty much every cell line we've looked at. So in addition to increasing ATP levels, in addition to enhancing glycolysis and OXPHOS metabolism, we see an increase in cell survival under nutrient deprivation stress when we get rid of AK4. So again, we have this model where we're building more responses in here to loss of AK4, but we really don't know how that's working yet. We have this nice big question mark there yet. I'm not going to have time to go through all of the data for other things we've looked at, but we have looked at other types of metabolism like fatty acid oxidation, which does occur. We've looked at mitochondrial biogenesis. A student in my lab, Ryan Mirez, had a great undergraduate thesis where she showed that mitochondrial biogenesis is enhanced when you knock down AK4, which is probably leading to some of this enhanced OXPHOS. We also saw an increase in redox buffering, which can be an output of enhanced glycolysis. We saw increased autophagy, which is the cellular response whereby cells can start to consume themselves. And we have just a little bit of data here. We have normal cells right here. Maybe you can see that on your screen. There's some pretty dim speckles here. And if you knock down AK4, you see lots of those speckles. Those speckles are all of these things called autophagosomes. There are these little double membraned organelles that help to recycle cellular material and give that material back to the cell for oftentimes bioenergetics. So we have an increase in autophagy. If you like Western blots, we showed that there's an increase in autophagy by Western blot 2. Here's your normal AK4 levels. If you suppress AK4 expression, you see a transition from LC3B1 to a lot of LC3B2, and that LC3B2 is considered the type of a protein that's involved in making all these speckles called autophagosomes. And we see an increase of phosphorylation of this protein called ULK1 or ULK1. When you knock AK4 down, you have an increase in phosphorylation. That's really the trigger for starting this mechanism of autophagy. So we see increased autophagy. I think I have slides here showing an increase in our redox buffering capacity as well. So again, the gray bars are all the control bars. And if you just take a look at cells normally and knock down AK4 and then measure either glutathione abundance or NADPH abundance, you see a bump up in total NADPH, a bump up in total glutathione. And glutathione and NADPH are both used to help the cell buffer lots of reactive oxygen species stress, lots of raw stress. So it helps in the redox buffering capacity of cells. But when you deprive cells of glutamine, which really causes cells to be redox stressed, then you see this massive upregulation in the amount of NADPH and glutathione in the cell. So if you give the cells some sort of redox stress and then knock down AK4, you see a really dramatic enhanced buffering capability of these cells. So here we show that when we suppress AK4, we get all of these different phenotypes, which are pretty profound. And again, they span many different cell types from many different types of tissues, normal cells, disease cells. So so the question we always have is what's the magic question mark right here? What's the link between suppressing AK4 and all of this stuff? And if any of you have been in the cell signaling field or the cell biology field, you might already know what the answer is. The answer is this protein called AMPK, or adenosine monophosphate protein kinase, which is a master regulator of cell metabolism and stress responses. I'm not going to go through all this here. This is just to show you that AMPK, this protein right here, does all of the things that we've looked at. It regulates all the responses that we've looked at. So if you activate this protein AMPK, you're going to increase glycolysis, you're going to increase OXPHOS, you're going to increase the cell's ability to buffer redox capacity, you're going to increase the cell's ability to survive under stress. All kinds of important things are regulated by AMP kinase. So that was the natural thing for us to look at. We said, does suppressing AK4 activate AMP kinase? So here we have AK4 knocked down in each of our 3 different medias: normal complete growth media, glutamine deprived media, or glucose deprived media. We have our control and AK4 knockdown conditions. And if we look at AMP kinase when it's phosphorylated at threonine 172, which is indicative of AMP kinase activation. So when AMP kinase gets phosphorylated at this residue, it's activated. We see a slight or a major bump up in AMP kinase phosphorylation, indicating that when you get rid of AK4, AMP kinase becomes activated. And I'm not going to show you all the other Western blots here, but we have Western blots for every single well-known responder to AMP kinase activation, and all of them, except for one, show that they are activated as well. So not only is AMP kinase phosphorylated, it's activating and phosphorylating all of its targets. So that was kind of our answer. AMP kinase activation is responsible for all of these different cell biological responses of AK4 suppression. So that was interesting to us. That's a paper that's being written up right now. That's what those students that we talked about at the beginning did. They really knocked the ball out of the park in all of these. But we wanted to push this one step further, so that the second part of the title of this talk was looking for new therapeutic targets. So we looked at the bioenergetic landscape, which means that we looked at how mitochondria, proteins in mitochondria, impact the bioenergetics of a cell. We drilled down on AK4, but now we also want to think about new therapeutic targets. So what can we do with this knowledge here? So we know that if we suppress this one protein, this mitochondrial protein, we'll have this big old increase in AMP kinase activation, and that will lead to all of these things going on in the cell: more glycolysis, more redox buffering, more biogenesis and OXPHOS, more autophagy, and in the end, all of these things contribute to cell survival. What can we do with this? So we started to look for diseases that might have the opposite of all of these things going on, and then say, hey, if we can get rid of AK4 in this disease state, can we rescue all of these problems in the cell? So our first target was this syndrome called Lee syndrome. It's one of these rare mitochondrial diseases that's characterized by insufficient ATP production and increased redox stress. And this decreased ATP production and increased redox stress is due to patients that have mutations in different proteins that are part of the mitochondrial electron transport chain complexes. So here's complex 1, 2, 3, and 4. These patients have mutations in one of these subunits of these complexes. There's many different subunits that give rise to Lee syndrome. I'm just showing you a number of them here. They're all highlighted in red. And when these mutations occur, that causes insufficient electron transport chain action, which means decreased ATP production. And if you know anything about what happens when you mess up the electron transport chain, you're going to know that there's increased reactive oxygen species floating around in the cell, so a lot more redox stress as well. So these low ATP levels in the cells of these patients and the increased redox stress in these patients very severely damages tissues, and it very severely damages those tissues that are particularly dependent on mitochondrial oxidative phosphorylation. And that happens to be those muscles associated with neuromuscular, those cells and tissues associated with neuromuscular function. So the patients that have this syndrome are usually diagnosed around 2 years of age. That's how long it takes for low ATP levels and increased redox stress to accumulate in these patient cells, about 2 years. And then all of these patients die within about 6 to 7 years. It's a pretty tragic syndrome. These patients suffer a lot. There's really no cure. They can be put on different diets that might alleviate some of their symptoms, but all of these patients do die of asphyxiation, around 8, 9, 10, 11 years old, because their diaphragm and their lungs just can't work anymore. So this is the first disease that we turned our attention on with AK4. So we thought, all right, we've got this protein that when we get rid of it, it increases all of this stuff right here. These patients have decreased function of OXPHOS. Let's see if we can increase the function of OXPHOS. So the first thing we did was to take our cells and we knocked down, using siRNA, those proteins that are associated with this disease. These patients have mutations in these 3 different genes, giving rise to altered proteins that cause those complexes not to work so well. So we just completely knocked out those proteins that are part of those complexes. And we saw that there was a decrease somewhere between 20 to 30% of ATP per cell in these cells. And that's about that pretty closely matches the patient's phenotype. They have somewhere between 20 to 50% decrease in ATP production. And then on top of that, we knocked down AK4. And in each of these cases, we rescued the total cellular ATP levels in those cells that had decreased NFS1, COX-10, or UQ-CRQ. So in a very, what we admit to be initial and preliminary model of Leigh syndrome, we were able to rescue the ATP phenotype, increased ATP levels. We also wanted to understand whether we could not only increase ATP levels, but increase survival. And in these cells, when you knock down any one of those subunits, you decrease cell viability by somewhere between 10 to 20%. And that's pretty close to what patient cells look like too. Patient cells have decreased viability anywhere between 15 to 30%. So we saw just below a 20% decrease in viability here. And when we knocked down AK4, again, we rescued cell viability, indicating that perhaps starting to consider targeting AK4 might be a therapeutic potential for diseases or syndromes that have low ATP and increased redox stress as part of their characteristics. And again, we're pretty certain, we haven't completely validated this yet, but we're pretty certain that in these cells, AMP kinase is activated, increasing all of these cell biological mechanisms that we've talked about here, eventually leading to cell survival. All right, so in summary, we've done this mitochondrial screen. We identified AK4, that as a protein, when we suppress it, lots of increased in ATP levels. We do want to also mention that there are many other proteins that we identified in the screen, and there are students, undergrads particularly, who are starting to pick apart those one by one. So we have this, what we call landscape of bioenergetic changes. What's going on in the mitochondria, lots of different pieces in the parts of mitochondria that we can look at and how those change when they're suppressed. We've done initial characterization of AK4, and we found that we can, in at least preliminary models of Leigh syndrome, begin to rescue the phenotypes, the pathological phenotypes of Leigh syndrome by enhancing cell viability through restoring ATP levels, enhancing OXPHOS, activating autophagy, and restoring the redox balance. And all of this is likely working through AMP kinase signaling. All right, and with that, I'll stop. And if there happen to be any questions, I'll be happy to take them. I've already gone through my acknowledgements, so we'll open the floor up there.
Thank you very much, Nate. If you have questions, please put them in the chat or raise your hand and mute yourself and tell us. There is one from Sebastian. So the absence of AK4 may allow cells to use alternate nutrient sources for mitochondrial metabolism aside from glucose and glutamine.
That's a good question. We need to do those yet, and I have that. So who asked that question? I actually don't have the chat box up, but whoever asked that question, great question. That's one of the things our collaborators have been pointing us into too. So you're right in line with our collaborators, and frankly, you're right in line with our manuscript reviewers as well. They're asking us, okay, you knock down this AK4, you can obviously alter the metabolic paths through which cells utilize fuel. So if you knock it down, are you asking your cells or causing your cells to be able to preferentially utilize other nutrient sources? In our first response to that was we've done one assay with a collaborator we've done it a number of times. We're pretty confident with it now, and we see that we don't think that it's altering preference for fuel sources like glutamine or glucose. It's actually simply increasing the rate at which all these fuel sources are consumed. So we definitely see an increase in glutamine consumption. At the same time, we see an increase in glucose consumption. And so we think that we're just completely increasing the rate at which these cells are able to consume these fuel sources, but we haven't directly compared that to plasma levels of pyruvate or galactose yet. So that's a great question, and well, we just got to do the experiment yet.
Thank you. That was Sebastian who
Okay, thanks,
asked
Sebastian.
that question. So Christian Lopez is asking, since AK4 is found in wild-type cells, how did natural selection select for something that lowers viability?
Yeah, so how do we reckon with this? So there's AK4 in regular cells. So does that decrease the viability of cells? And the answer to that is we don't know if that's the correct way to think about it. It might be, but we don't know if that's the correct way. So let me see, I think my slides are still up. You can still see them. So I'm going to go back up and show. Okay, sorry. Here we go. Next slide. They're right here. Okay, so this is what we think we're seeing. We see a little bit of an increase, a little bit of a bump in viability, or this is glutathione abundance, this is NADPH abundance. We see this is the same thing holds true with viability. I think maybe on the previous slide or one of the next slides, I don't remember where it is. We'll just have these as stand-ins. There's always a teeny bump in viability or glutathione abundance or ATP in normal conditions. But when cells undergo stress conditions, that's where you see the big increase. So we don't actually see a decrease of viability when we have normal levels. And although I'm not showing the data, if we overexpress it, AK4, we don't see a decrease in viability. What we do see is that the cell has this big increase in the ability to respond to adverse situations when they're in adverse situations. And so when they're stressed, then we see this big response. I don't think I've completely answered your question yet. It's a good question. The best thing we can do is to continue our overexpression studies. And what we've seen so far is that it's actually been pretty underwhelming when we overexpress AK4. So we put even more AK4 in, we really don't see any effects. So it's a good question, but we don't think that there's actually a decrease in viability or a decrease in anything when we overexpress it. It's just the opposite is true. When you decrease it, that's when you see the effects. And the cells can turn on and off AK4 expressions under different conditions. So when cells are naturally stressed, there are I don't want to say collaborators because they're not technically collaborators. I would say they're more competitors. There's competing labs who have shown that when you stress cells, they then suppress expression naturally by methylating the promoters of AK4 and decreasing AK4 expression naturally.
Thank you. I think that Melvin has a question. Go ahead.
Yeah, so I was just, first, wonderful talk, but I was just wondering, what do you think is kind of like the direct mechanism between AK4 and AMPK? How is it regulating AMPK? Do you have any thoughts on how it does that?
Yeah, we have some thoughts. If we can prove it, we can finally publish it. So, so what we see is that when you knock down AK4, all adenosine nucleotide species go up. So I've been focusing on ATP here, and it's true, ATP goes up. So does ADP, so does AMP. All of the adenosine nucleotides go up. And what we've seen in some initial characterization is there are these metabolic pathways called nucleotide biosynthesis and nucleotide scavenging pathways. And we see an upregulation in the expression of genes or proteins involved with both nucleotide scavenging and nucleotide biosynthesis. So probably what's happening is when you knock down AK4, nucleotide scavenging, nucleotide biosynthesis go up, and that increases the level of all of these adenosine species, okay? But when you increase AMP just a little bit, AMP is this really strong trigger for AMP kinase activation, AMPK. So AMPK is going to respond just to that one nucleotide species that's increasing AMP. It's going to be activated, and it's going to cause all these downstream effects that we've talked about. This is something that we've been trying for a while to demonstrate. We have some, what I would say, very solid preliminary data. It's pretty technical to do these assays, so it's a little bit cumbersome. It takes a while. And honestly, we're trying to show it. But that's our best guess, is that all nucleotide species are going up, and the AMP species particularly is activating AMP kinase, leading to these downstream effects. Good question.
All right, Sebastian had a couple questions more. I'm going to summarize them both. Whether you have measured the level of ROS upon silencing of AK4, and whether a phenotype of extreme viability that can be associated with cancer cells, for example, will be related with the AK4 maintaining that hyperviability.
Yep, great question. So first of all, yes, we've done the ROS experiments. Our results have been underwhelming. We haven't seen a ton of ROS increase. Definitely nothing statistically significant. The best I would say is trends of increased ROS, and that's probably stretching it and looking at it with my biased eyes. Other labs have published two different results. One set of lab consistently publishes an increase in ROS when you knock down AK4. The other lab consistently publishes a decrease in ROS when you knock down AK4, and that would match more with our experience, a decrease in ROS. The other labs, I believe their data. I've reviewed their papers. The data is sound. The best we can say, it might be cell type differences, and all of their experiments are in unstressed conditions, while we're doing our experiments in stress conditions. So I don't have a resolution to that answer yet, but it's a great question. The second part is, what's going on? Is this going to contribute to cancer? It very well might. So if you take a look at AK4 and look at AK4 in different cancers, AK4 is commonly mutated in many cancers, and it's mutated right in the enzymatic domain. So it's mutated right in the domain where this thing that's required for this thing to have activity. So many cancers have essentially a dead AK4, and this dead AK4 might be contributing to their ability to respond to redox stress and to enhance, in particular, glycolysis, because we know that glycolysis is really necessary for cancer cells to crank out all these biosynthetic precursors. So there are a few cancers where it's highly expressed. And then this other paper that I was talking about that I didn't have time to publish today, or talk about today, that we just got accepted this week, is demonstrating that out of the 9 adenylate kinases, AK4 expression positively correlates with lung cancer progression from stage 1 to stage 4. It very tightly correlates with expression. So yeah, there's lots to think about there.
Thank you. I have a question that I kind of like presume the answer. So are there any inhibitors of AK4.
I wish there was, and that's the dirty secret. I didn't even tell you guys that. So I just talked about AK4's active domain and enzymatic domain. So AK4, there's actually not much published on it. There's 50 papers, something like that. And the biochemists who study the activity of AK4 are completely divided. They are divided between saying this is a naturally dead adenylate kinase, and this is no, it's actually not an adenylate kinase, it's a GTP, it's a guanylate kinase. So there are no inhibitors because the field can't even determine whether this thing is an active kinase or not yet. So unfortunately, no, I would love it. And if there are anybody whoever comes across one, let me know so I can drop this drug on these cells and see what happens, because that would be obvious, right? If we're talking about therapeutics, it's an enzyme, it should be druggable. So let's do it. Yeah, good question.
Thank you. Any further questions from the audience?
I guess kind of just regarding the inhibitory question, is there any like structural information of this protein at all?
Yeah, there's good structural information. So the labs who argue back and forth about whether it's an active or inactive kinase produce some crystal structures, 12, 15 years ago. They're not great resolution, but it does demonstrate. I actually probably fall in the camp thinking that this thing actually might be a dead enzyme. So there's a typical adenylate kinase domain. It's very well conserved. There is one mutation in there that might render this thing dead. And then it has a lid domain. It's kind of an enzyme that has to have a lid closing on top of it to trap the nucleotide inside. That lid domain is actually heavily mutated in cancers as well. So yeah, there's some okay structural information on it. And yeah, if we're going to design some drugs, that would be where you would start to.
Thank you. If there is no further question, we thank Dr. Lanning again. And thank you everybody for your participation. Marcelo, you want to do the closing?
Yes. Okay. Yes. Thank you, Veronica. Well, this is the moment when I want to thank everybody, thank the speakers, the poster presenters. Especially, I want to say thank Yvonne, Veronica, so the whole organizing committee, that is Dr. Ramirez and Dr. Jimenez, but Veronica Jimenez in particular and Yvonne are the brains behind the Zoom Slack operation. So without them, this would not have been so smooth. So I have to thank you especially. Thank you, Ned. Thank you, David, who is not here. I think he already disconnected. We had, I think, a very good CABSCon6, and I hope, Nate, we will see you in person in CABSCon7 as a participant, and all of you. Let's hope that this is our final online CABSCon, and next time we go back to normal. Okay, thank you, everybody, and this concludes the 6th edition of CABSCon.
Thank you.
Mm.